I'd like to welcome you to today's presentation of the Congressional Biomedical Research Caucus. Our briefing today is featuring Dr. Regina Armstrong from the Uniformed Services University of the Health Sciences. I'm Joan Goldberg, Executive Director of the American Society for Cell Biology. We're a member of the Coalition for the Life Sciences, which helps support these caucuses. I'd like to thank the co-chairs of the caucus, Representative Brian Bilbray from California, Representative Charlie Dent from Pennsylvania, Representative Jackie Spear from California, and Representative Rush Holt from New Jersey, all for their commitment and ongoing support of the caucus. I'd also like to thank the Howard Hughes Medical Institute for its grant, which helps support these timely and important briefings. We videotape every briefing, and if you can find past briefings on the CLS, Coalition for the Life Sciences website, www.coalitionforlifesciences.org. And if you'd like to register for the RSS feed, then you'll learn when the next posting will be. Dr. Armstrong has served as director of the Center for Neuroscience and Regenerative Medicine, CNRM, since 2008. The CNRM is a collaborative intramural research program at the Uniformed Services University of the Health Sciences, or USIS, that is in collaboration with the National Institutes of Health and the Walter Reed National Medical Center. And she'll talk a little bit about that collaboration because all of you who work in government know that collaborations across agencies are not always easy, but are really important. She directed the translational research program of the Defense Brain and Spinal Cord Injury Program from 2002 to 2008, as well as the Comprehensive Neuroscience Program, 2003 to 2006. She's also served on the Oversight Committee for the Blast, Lethality, and Injury Program, 2008 to 2010. Her research efforts in her lab have been funded by grants from the NIH and the Department of Defense, and her work is focused on the regenerative potential of endogenous neural stem progenitor cells in rodent and human central nervous system models, and she can explain that a whole lot better than I can. She hopes that her work will prove helpful in combating the consequences of traumatic brain injury, or TBI. TBI afflicts between 10 and 20% of troops returning from Iraq and Afghanistan. In addition, more than 1.1 million civilians each year seek emergency treatment for TBI. She'll discuss research into treatments for TBI, issues related to diagnosis, and also affecting some long-term consequences from that initial trauma. Regenerative medicine attempts to improve recovery by inducing plasticity in the brain, by stimulating repair, and by exploiting stem cell transplantation. Also by modifying the environment of the damaged brain region, the area that's been affected. Please join me in welcoming Dr. Armstrong. She'll take your questions at the end of the presentation. Thank you.
So I'll talk a little bit about this center, just a few slides, so that you understand this collaborative interaction going on across federal agencies. And then I'll try to introduce the issues of traumatic brain injury and diagnostics through the repair aspects, and branch that into the regenerative medicine topic, and try to explain a little bit about regenerative medicine approaches overall in the central nervous system, but specifically the challenges of applying those to traumatic brain injury. So this is the center in terms of the organizations involved. So our congressional language, it's actually a federal intramural program. Our congressional language actually pairs these three agencies together, or groups together. So this is the combined, what used to be the Walter Reed and National Naval is now combined into this new entity. It's right across the street from NIH, as are we at the Uniformed Services. But this is Navy, Army, and we're DOD, but not really Navy or Army. And then this is Health and Human Services intramural program. So we sort of joke that like any federal rule there might be for biomedical research we have to deal with. But it's worked out very well. This side of the organization that is my co-director is Walter Koroshetz. He's the deputy director of the Neurology Institute just under Story Landis, and he's an incredibly wonderful partner. He's a neurologist. You might have seen him on Sanjay Gupta and all kinds of famous things. He knows his stuff, and he's a clinical guy as well. So we have a really strong partnership with NIH intramural. And with the Walter Reed Navy, at the Uniformed Services, our clinical faculty, this is where they do their clinical work. So just to give you an idea of where we are, the center is basic, translational, and clinical research into traumatic brain injury. And we deal with military and civilian patients. And I'll just mention that for a little bit because, I mean, the reason we were given the opportunity to start the center was to help the military patients, and specifically the Walter Reed and Navy patients. But we need civilian patients for a couple of reasons. One is that it's very hard to do very early stage research in theater. So where the patients from the military side are actually receiving that initial TBI insult, you can't get to them in 48 hours with the same kinds of things we can do with the civilian partnerships. And the NIH actually has magnets, 3 Tesla MRI magnets and setups to do research at some of our local hospitals in the Bethesda area. So we can do that. We can get some things done within the first few hours like you need to for acute injuries. So it's a great partnership in that way. The military patients who enroll in our studies, they can come to studies going on at the hospital here, the Walter Reed Navy Hospital, or they can come into the NIH Clinical Center, where we've developed quite a bit of facilities there specifically for these studies. So the military can go either place. The civilian can't really be seen here, but they can go to NIH. So it's a really good partnership for helping to expand the range of work we need to do. And as anybody who's done any clinical studies knows, recruitment is the highest bar and hardest issue to deal with to really get done what great ideas you might have. So this is our goal. It's really to improve recovery from TBI in the military service members, but to do that, we're working with the civilian as well. And the first thing that I'll talk about is what we need to do to characterize the injury. And that doesn't sound like a big deal, but I'll explain why it really is. And we need to know that to really do an optimal treatment plan from what we have available now, as well as what we're trying to develop. And we need to be able to evaluate outcomes. That's really a challenge in this field. I'll explain that a little bit, to know what really is or isn't working properly. And our regenerative medicine definition is really any repair strategy. If we can improve any function in any soldier, we're happy. Service member, sorry, not soldier. So for neuroregeneration, we sort of divided that term up to be anything to do with making new cells and new processes from those cells to try to connect. For neuroplasticity, that means a lot of things to a lot of people. In our terms, we're talking about the new synapses that help the neurons connect with one another and the circuits that those make and how those circuits function. So we'll consider that a regenerative process as well. And then rehabilitation is a big part of anything with traumatic brain injury. And that's really what you need to do to bring functional capacity online for day-to-day movement issues, cognitive issues, really function. Neuroprotection I won't talk about a whole lot, but it's really obvious that it's better to save a cell and its connections than it is to try to go through all of what I'll talk about to replace them, because that is really difficult to do in the central nervous system. So in the brain, we would really like to limit the initial injury and how that makes further damage through secondary cascades. So that's a very large field of not necessarily regenerative medicine, but making a better recovery. So in traumatic brain injury, I'm not sure how many people realize that there's a lot of overlap of symptoms with post-traumatic stress disorder that we hear a lot about as well. So this is my simple-minded schematic of things that can happen in terms of the range. If you have a really severe traumatic brain injury, you know that. It's not a question of whether you do or don't have that. But when you get into these like less severe versions where I've made it this little triangle tapering, that mild traumatic brain injury is actually much more difficult to know what level of injury you have, exactly what's affected, and what the long-term outcome might be or not for that particular patient. Post-traumatic stress can be sort of the other way around. There's certainly very severe post-traumatic stress that's not combined with traumatic brain injury sometimes, and you really have that diagnostic very down. But there's also very complicated cases and milder cases that make it much more difficult. And there's a lot of what we call comorbidity, where you have both together, to the point where some studies are actually hard to recruit into the separations that they're trying to recruit into to do those studies and differentiate them better. In the military, we have this underlying scenario to deal with where those patients are in conditions of heightened anxiety for prolonged times. They have fear and arousal issues going on, a lot of endocrinological changes, and they have a lot of frequency of repeat exposures. So they might have a blast and get thrown into a wall. They might still be okay after that. They might still be in theater, have another one. You can get these repeat injuries for different reasons. And that's true in the civilian population as well, and the sports injuries that we hear a lot about. But it's a concern in the military for sure. So when we talk about this mild traumatic brain injury, then you start to talk about what's actually 70 to 80% of the numbers you hear. So when you hear, over 1 million cases in the CDC numbers, 70 to 80% of those civilian are mild traumatic brain injury. That's also true in the military. The initial insult, the initial TBI, 70 to 80% are going to be mild. That doesn't mean that they don't have chronic symptoms. It just means that the extent of damage by the criteria is not higher than the other ones. It's considered mild, but they can still have chronic symptoms. And those people that have progressed to chronic symptoms are called the miserable minority. And they're very hard to distinguish from the initial insult and know which are going to go have chronic symptoms the rest of their lives and which are going to recover completely. And anybody that's done a drug trial of any kind can tell you, if you have a large population recovering, your drugs are going to fail. So we need to be able to know who are the ones that are not going to do well, and that's a good risk to treat and a good chance that we'll find the effective treatments. So you'll hear a lot about diagnostics in my talk because of that. The other group that we mentioned a little bit here is this moderate TBI. When you get into that middle ground, you actually have a little more complicated injury as well, in that you've got gray matter out in the brain where the neuron cell bodies are. Those get damaged. They don't recover very well. They don't get replaced very well. And you also have white matter tracts, where those are the connecting parts of the brain, right? And those have damage to the axons that are the connecting parts, but they can be very diffuse throughout the brain in a typical moderate types of TBI injuries. So if you have a very diffuse injury throughout the brain, that makes it harder to treat and identify what's really going on with that patient as well. This, you would think, okay, so those mild or moderate ones, they're a little harder to figure out. The severe, they must be easy, right? Well, this is an example of the severe. I don't know if you can see these CT scans well enough, but these are the types of images that are standard of care when a patient goes to the emergency room. They get a CT scan as part of their workup. These are what we will call severe TBI by the criteria that neurologists use, the Glasgow Coma Scale. You can see these are very different. I won't get into the actual, the neurology and the radiological terms, but these are very different injuries, one to the other. This is the diffuse axonal injury. There's problems throughout. You don't see it the same as you do with this contusion. And each is a different set of symptoms and a different set of damage that goes with it. So that's hard, even for the severe. And then what's going on is actually very different pathology in terms of the hemorrhaging and the axonal damage or the contusion. They have very different pathology. So we actually need to be able to figure out not just where something is happening with the imaging, but what is it that's the underlying pathology, because you need to use different methods to repair different damage. So this is an example of where we need to know exactly what's damaged and where it's happening and how that fits the symptoms. And this is a very good example that Dan Pearl lent to me. So he's a neuropathologist that we recently recruited to the university specifically for this center, because we knew a big black box in the military is, with these repeat injuries that are going on, does the military have the same problems we're hearing about in the National Football League and those repeat injury cases we hear there? We're getting a lot of information, and this is an example of one where these patients, the older, not even, you'd like to think about it as the older NFL players, but actually some of these players are young. One of them that's sort of a really tragic one that gets to my heart is one that was an Ivy League player at UPenn, and he actually committed suicide, and he had some of the things I'm going to show you in these older patients that had a lot of injuries. So what happens is that this blue is just showing you where all the neuron cell bodies are. This is the white matter in here, and the brown is actually staining for a problem that's developed by accumulation of what's called phosphorylated tau. So this phospho-tau stain shows you the degenerative aspects. And this area of the brain is very involved in emotional responses. It's very involved in memory. And what this player had was he had problems speaking as well, which you can't really tell from this quite as easily, but problems speaking, problems with attention. This is very common, outbursts of anger and memory problems. And these are all connected. There's a lot of connections to the frontal areas of the brain that control a lot of these impulsivities. So it's very widespread. You can see it in these different areas. And it often hits these memory and emotional areas as well. So this tells us a couple of things that I'm trying to bring forward, in that this chronic traumatic encephalopathy, that's now being developed in terms of understanding it as a neurodegenerative disease. And this is traumatic brain injury causing some of this. The connection is not that clear yet. We need the research to really prove that that connection is due to this and that these symptoms are due to this. But that's what we brought Dr. Pearl to do in the military, and he's been collaborating with the group at Boston University under Ann McKee, which is the group doing the Sports Legacy Foundation and trying to accumulate the NFL cases to really try to pull these pieces together and say, what are we really dealing with? Because it's still a bit of a black box. So for the military, we need to know across a spectrum of TBI. So in the military, we have pretty much the most diverse type of traumatic brain injury you can imagine. There's these blast waves that we don't really even know yet exactly what they do or don't do to the brain itself. There's concussions because when you're in a blast, you get thrown. When you're driving around, you get in accidents. The military do that just as much as anybody else. The whole Humvee gets thrown. The body gets thrown and then stopped very quickly, causing acceleration, deceleration, so the brain just goes back and forth. And then there's penetrating injuries like shrapnel and things like that if they don't have their helmet on. The military has started to coin this term blast plus, which means they have a blast exposure plus many other things going on. It's not just a blast exposure. Of course, in the military, as I mentioned, repeat exposures are a big issue. And what the context of high anxiety and stress does to how that patient responds. Does it make them more or less likely to develop post-traumatic stress simultaneously? Does it make their recovery different because of all the changes going on with the stress events? So we need to know the damage. So one of the things we're doing a lot is trying, in the field, not just a center, is trying to improve diagnostic tools. And some of those will be imaging tools. And then there's other aspects like what we call biomarkers, which means it can mean a lot of different things. What people think about often is they take somebody's blood and you look for certain molecules in it, and that tells you something about what's going on in the brain. So we want anything we can do non-invasively for diagnostic tools. And if we can develop these better, then for a given patient, we can do early detection. And we can also do what I mentioned earlier, which is focus what are the selection criteria for a given treatment plan to match that patient and make it more likely to be successful and for us to know whether it is or isn't successful. And we need that kind of combination of pieces to come together in order to develop new interventions, because we can develop interventions then based on the mechanism of damage and have a much higher likelihood that we'll get the right patients, we'll do the right treatment for that patient, and it will have a higher likelihood of being successful. So for those who aren't quite so familiar with imaging, this is some of the state of the art. I don't know if you can see it quite so well, but this is a version of magnetic resonance imaging. And this is non-invasive. It's just done by water molecules moving in magnets. And these three colors tell you what direction fibers are going in the brain. So you can tell a lot about this and all the fiber tracks. So as I mentioned, the connections from one part of the brain to the other often get damaged in this diffuse way. And you can actually look at very specific fibers and track sizes and whether they are or aren't connecting the right numbers across very specific areas and do that kind of analysis. This version of MRI is called diffusion tensor imaging, or DTI, and that's really important in the field right now to try to look at the fiber tracks as well as the neurons themselves and the overall structure. So the connections are big. The other part that's really big right now is what's called positron emission tomography. And this helps us to see not quite the level of detail, but it tells us metabolic information. Like this is a glucose being used. There's a big alteration of glucose being used when you've had a brain injury. And if the blood flow that you see in the MR and the glucose that you can see in the PET, if this doesn't sync, you have the wrong things happening around the tissues and cells die more than they might otherwise. They don't recover, that kind of thing. So we want to know a lot about the metabolism, and we can do that with PET imaging. And then there's a lot of other molecules you can probe with PET imaging if you design radio tracers to do that. So you can look at transmitters that connect and send the signals. You can look at the barriers that protect the brain and how intact they are or aren't by specific things you put into the patient to see them in this kind of PET imaging. And the center, I won't really talk about this, but I'll just mention it and you can ask later if you want. The center has the second in the country ever the combination of these two into one machine to help register as best we can exactly what's going on in a particular area and get this kind of resolution paired up with this kind of metabolic information. So it's made by Siemens. It's called a molecular MR. The first one installed was at Mass General in Boston. The second one is just coming online at NIH today, and I think the release will come out tomorrow, so don't get me in trouble. But I thought it was very important to mention today. So the mild TBI, I mentioned a little bit about the significance of that population because it's 80% of the total TBI incidence you'll hear. So in terms of total TBI incidence, one point I want to make is the military and civilian numbers overall are somewhat surprising to some people. So this is Defense and Veterans Brain Injury Center, and they keep track of the US military traumatic brain injury, and they're tracking about 28,000 service members a year get a TBI. The CDC tracks the civilians in the US, and they have about 1.7 million and calculate about twice that many living now with permanent disability. So that's a lot of people. And if you think about it, this 28,000 coming back each year in the military, they're not going to go away if they develop chronic symptoms either. So this will tell us it's probably going to be similar in the long run to what the military will have in their chronic exposures to this. So we need to develop tools not only for the acute cases that can be treated within the first hours of injury, but how can we diagnose the ones that have had the chronic symptoms, know that they're due to a specific mechanism of TBI, and how do we pick a treatment or develop a treatment to work for that patient? One of the things we're beginning to be able to do is to help identify a little bit better what's going on with mild TBI and some measures to identify those patients that might have trouble. And this is work from Ramon Diaz-Aristia. He's another new faculty member we just recruited to the center. He's a neurologist who will help with some of the clinical trials we'll do in the next 6 months to a year. So he's developed a term and a way to identify what we call complicated mild. So complicated mild TBI means that they're not doing so badly based on the neurological score that they get, but they actually have symptoms or signals that you can see on that initial CT that you get in the ER. So they're called complicated mild because a lot of mild don't have any CT findings. And then you can also combine that with the MR that I just showed you and look at a couple different ways to identify volumes of lesions on MR. And if there's a certain volume of lesion on that mild patient, they may be more likely to have problems long term and less likely to recover on their own. And that gets combined with neurocognitive assessments. That's a whole battery of those. So if you take those three things together, the CT findings, the volume of lesion that's identifiable by these other new techniques that are not standard of care, and these neurocognitive assessments that also are not standard of care, you can actually identify a cohort of patients that we're starting to study now and say, do those really represent the ones that are going to be chronic and be the miserable minority long term? Are those the ones that we should be able to try for certain treatments and see if we can prevent those long-term symptoms? So we're doing these studies to try to identify how do these initial findings, when somebody comes into the ER, how do they predict these long-term consequences? And you have to do longitudinal studies for that. The Congress mandated the Defense and Veterans Brain Injury Center to do a 15-year study, which is a really long time to do a study for some things, at least for traumatic brain injury. So that's going on now. They're only in their first legs of that. And those are called natural history studies, and they're being done in the military for that one. And we're partnering with the Defense and Veterans Brain Injury Center to do that. And we're also doing a civilian cohort at the NIH Clinical Center. And the idea is that we need to take this imaging that we're talking about and these batteries of neurocognitive tests and emotional tests, a whole range of tests, and really try to identify how do we tell which patients need what and does something work or not, so that we can really get to the chase and get some treatments that we can test for real and have an answer. And right now, we don't have drugs that are going to work for this. Lots of people are testing drugs. Lots of drugs worked in animals and didn't work in humans, probably because the animal models don't do as well as we might like in mimicking the injury, or enough animal models weren't tested. But also because of exactly what I already explained, the humans didn't do well enough in this deal. The mild TBI was too hard to tell which ones were going to recover. So it made it very hard to do the type of analysis you need to know what might actually be working. So if we could come back to some of those treatments and try them with the right cohort of patients, we might actually be able to know which ones are working for what type of injury. But right now, rehabilitative medicine is what we are really focusing on as well, and continue to do to improve function. So this is really the major option right now for TBI to promote recovery. What we need in that field is really rigorous studies to optimize them. They need us to be able to differentiate among this heterogeneous injury context. And they really need to be able to understand more what we call patient-based factors. So that doesn't mean a whole lot, but it refers to a lot of things that explain why does this patient respond this way and this patient not respond that way. You hear about some incredibly good recovery stories, and you just wish, couldn't you figure out why that one that did so well? Can we study that one and figure out how to apply it to this one? And there's a lot of factors in people's attitude, their support system, their genetics, their epigenetics, and a lot of these things that you can go on and on about, but you need to be able to figure out, are there key ones? We know support system's huge, we know attitude is huge. What are the other factors that really play into the success or the lack of success for a given patient? So we need to understand these neural substrates that go with the rehabilitative medicine so we can make it more directly mechanistic and apply it in the right people. And this overall health and mood, of course, it plays into your neural substrates and your plasticity most likely. But we need specific strategies for specific functions. Not everybody has the same tracks in their brain damaged. Not everybody has the same part of their cortex damaged. So you can't do a one-size-fits-all. So rehabilitative medicine needs more specific haptic devices and mechanistic devices that can really, like, say for this patient, this one is what they really want to focus on. That will bring them online for the most bang for their buck, essentially. And then there's these so that ties into the plasticity where you're trying to get neurons that are surviving to make better connections if they've damaged those. You might be trying to get neurons on one side of the brain to take over for the other side, things like that, or adjacent areas. So the synaptic plasticity can take some surviving cells and make better circuits for these functions. So there got to be the underlying factors, but we don't really know enough about how this works to tie it in at a mechanistic level and say, for this drug development, this is why this didn't work. It didn't get put with the right rehab for that plasticity to make those come together, or it didn't induce plasticity. It only induced new cell bodies. We have to understand all the mechanisms to put them together, and that's also the cell replacement. Sometimes you need new neurons. You don't have enough of them. You don't make new ones for the majority of your brain. The glial cells can be the same thing. Those are supporting cells in the brain. They're a little bit easier to replace. Maybe a lot easier to replace than the neurons. But replacing cells in the brain is challenging. So that gets us to what I was asked to talk about, which is regenerative medicine. So first I want to just, in the big picture, really broad strokes, give a little bit of a view of why is this hard to do in the CNS compared to a lot of other tissues. Like there's another group that was funded around the same time that we were to set up the center called the Armed Forces Institute of Regenerative Medicine. And they're growing new skin for scarring. They're making new bladders grow. And all these things that I just wish we could do in the brain. And the brain just doesn't do it that easily. I mean, not that that's easy, but it's really a challenging environment to try to do it. So why is that? So there are actually neurostem and progenitor cells in the adult human brain. We don't really know for the adult human how many and what all their capabilities are. We do know in rodents that the adult can do a lot of things. So these are what we call the endogenous cells. These are what's surviving, what's in the brain, endogenously persisting, whatever word you want to use. So there are some there. I don't know if you can see it, but these are some of the markers we use to identify them in the brain or in tissue culture from the brain. The other thing that's going on, though, is when you have an injury, there's signals that inhibit the recovery. So if these cells try to do something, they get blocked by some of these other factors and molecules that get made in the injury site. And then there's these complex connections. So I don't know if you can see it very well, but this is one of those connectomes of the brain, which tells you where all the different parts connect as they're trying to signal. And that's pretty darn complicated. So we've got to get a way to have a handle on what are the most important connections for a given patient that we can try to bring online so that they could, move a certain part of their body if that's what their problem is. We don't necessarily have to fix all of them. We need to fix certain functional ones, a little more of a prosthetic kind of thinking than a total new brain kind of thinking. So those are some of the challenges. And this is, at least for the center, this is the approach we've taken, which I think reflects the broad approaches going on within the field, which is we want to stimulate whatever the endogenous cells can do. So it's not that they can do everything you want them to do, but we would certainly like to develop treatments, rehabilitative medicine plans, whatever we can, that will make them do as much as they can. Because for some patients, transplanting is not going to be a good idea. That's too risky for somebody that has a certain level of symptoms. So you have to do a risk-benefit ratio. And we want to get the endogenous situation to do everything it can. So we need to stimulate them, and then there's this transplantation of what we call exogenous stem cells. There's a number of different populations that can do that. I'll talk about that in a minute. So the idea with that is when you do need to replace lost cells. But one of the things that's become fairly well known in the field in the last couple of years is that these cells do more than just try to replace lost cells. They actually have a nurse effect, which in scientific terms we call paracrine, but nurse seems to work better. So they have a nurse effect on these endogenous cells. So when we put these in, they actually stimulate these better than most of the other things we try. And then there's these inhibitory factors I told you about. So we want to modify those. I think Stuart Lipton in the field used to call these PATs, or pathologically activated therapeutics. And the idea is that with your brain, you can't handle side effects of drugs. So if you put something in as a pharmaceutical that does something to everything in the brain, you have a high likelihood of side effects. If you can identify something that's upregulated just in that lesion, then when you put your drug in, it might only affect the area you try to affect. So it's a different way of getting a targeted effect by just understanding the context of the lesion and what those factors are. So that's focusing on modifying the lesion environment and what's inhibitory to repair. So this is my way of trying to explain in broad strokes a progression within the field of how we came to where we are now from some of the early studies, and how this nurse effect was identified, and which diseases are some of the target ones that are seeming more amenable in the CNS. For every level that I'll talk about, there are dozens of studies that I could have used as an example, and I'm just going to pick one to help with the progression. So one of the early studies being done were studies related to multiple sclerosis, or MS. And preclinical or animal studies of those they were transplanting in neurostem cells. And what they were trying to do was a fairly low bar, which is why the field was MS field was very popular for some early stem cell studies. And that low bar is that the neuron cell body is here, its axon goes here, has to connect to, like if it's in the brain, has to connect down to the spinal cord, and that's a pretty long distance. And they're covered by this insulating sheath called myelin. And there's a glial cell type named the oligodendrocyte that makes that myelin. If you lose that myelin because of something like multiple sclerosis, which is an autoimmune disease, the immune cells come in, they attack it, your myelin gets trashed, that neuron now can't signal and connect. So it's just like it wasn't there. So all you need to do, at least in theory, in the multiple sclerosis case would be if you can get that myelin back on, those will connect and maybe start functioning again. And it's a good proof of principle that this works, and you could bring functional recovery to some of the patients. So it was a really good place to start some of these studies. And that's actually the field I was in and still am in sort of more before the TBI work. What happened though is that because people were using an autoimmune animal model, when they put in these neural stem cells and they looked for the intended cell replacement that I just mentioned, they actually got a lot of modulation of the autoimmune disease. So the overall disease severity went down, and the animals just didn't have as much of a severity of a disease course. So the investigators realized this modulates the whole immune response, and this could be a really important aspect. And they also started to notice that those endogenous cells, we knew a lot about these neural progenitor cells in the context of multiple sclerosis models. So they knew how to identify those in the tissue, and they could then tell that, oh, these neural stem cells are changing the immune response and stimulating these endogenous cells. So that was what we call the paracrine effect, or the nurse effect, and that became a really big deal in the field. And other models like ALS studies started to bring on some of the same kind of information at the same time. But this is just one example from Stefano Placino. So then the MS clinical trials started to go forward, and it became clear that lots of types of neural stem cells could have this effect, not just lots of types of stem cells, I should say, not just neural stem cells. So mesenchymal stem cells, or MSCs, started to be used because they could be taken out of the bone, cultured for the specific patient, and potentially put back in. There are a lot of logistical reasons and immunological reasons that they were better. So they were put into MS patients and begun at several locations for clinical trials, really intending for this immunomodulatory effect and a potential effect on the remyelination and protecting the axons. So these started to go forward in terms of human studies. Stroke trials started to go forward as well. Now in stroke, you have a lot of the similar damage that you might in traumatic brain injury, but it's a little easier to figure out exactly where it's affected. You can tell what course that blood vessel went that had the stroke, and it's a more delimited territory. So that was sort of an amenable place to try some of this because you could actually identify when you got recovery, when you didn't, and try to look for protective effects. And some umbilical cord cells, umbilical tissue cells started to be used for that as well as many other options. And that became amenable as well to this intravenous delivery, so you didn't actually have to put them right into the brain or the tissue you're looking for. So for immunomodulatory, those looked promising. The problem with these is often that the intravenous delivery would often the majority would get stuck in the lung and the capillary beds of the lung, but you can still get the immunomodulatory effect. And then you try for this nurse effect and look for it whenever it might happen. And then a lot of you may be familiar with the spinal cord injury studies that have taken things a step differently. So these are trying to directly transplant into the spinal cord. This is a study that's going on now by Geron. And what they did was they took embryonic stem cells, and you don't want to do too much with those directly because they're going to make tumors. But what they did was they differentiated them to these oligodendrocyte progenitors, basically to the stage they were looking at up here. And they're trying to do the same thing. They're trying to get this remyelination, replacement of oligodendrocytes, and potentially also this nurse effect. But in this case, they are directly transplanting right into the spinal cord injury. And they're using this oligodendrocyte stage to try to do that. So this is sort of a progression of how you can see some of the fields overlapping and bringing different stem cell regenerative approaches forward for each different population. And it's a little bit hard to keep track of what cell type people are using, and I'm not going to go through all of them. But big picture, the embryonic stem cells that are being used for CNS diseases, they're pluripotent. They have a high potential for cell replacement because they can become anything. But they can also develop tumors that we call teratomas. So you have to be very careful about how those are used. Usually they're differentiated along a specific lineage or cell type that you want to have the cells become, and that is done to a certain stage of maturity before transplantation so that they don't develop tumors. And that's sort of what the Geron group did with the differentiating them into oligodendrocytes. There's induced pluripotent cells, which are ways to take cells and reprogram them to this pluripotent state by putting in certain genes. And if you, these are very popular right now for specific genetic diseases where you can take that genetic disease, you can reprogram the cell. That cell will still have the genes that came from that patient, and you can try to understand more about the mechanisms of that genetic disease, things like ALS and whatever. I mean, there's a lot of neurodegenerative diseases people are using those for. And there's a lot of other IPS purposes too, but I'm just talking about the ones that are most relevant to the CNS diseases. There's induced neuronal cells, and those are similarly reprogrammed, but they never go back to their pluripotent state. They just go directly from being like a fibroblast, which is a mature cell, over to a neuron. So those seem like they're interesting because they're less likely to cause tumors, but the problem is that without this intermediate stem cell stage, you can't amplify them up enough to get enough cells to do the transplants. And then there's lots of different stem cells from all diverse sources, umbilical cord, bone marrow, fat, and there's transdifferentiation effects where you're trying to make them become a certain cell type. And then there's those nurse effects or paracrine effects. And they all have different advantages and disadvantages for each of these, depending on what you're trying to do and where you're trying to put them. And so I can't really get into all the details of that, but regenerative medicine is a really big field right now. So a couple of the considerations for TBI and the CNS that I'll sort of end on the last couple of slides. So for long-term considerations, when you're talking about cell therapy or transplantation in the CNS, is the tumor formation that I referred to. There's also concerns in the brain, especially when you're talking about cognitive, neurological, psychological. You really don't want to cause dysfunction. So all those connections that I told you are so critical, and all the plasticity that we're trying to make happen, we don't want just any plasticity or any connections. We want the right ones. To some extent, the rehab and the cognitive aspects, you expect they might sort of prune to the right things once they're started. And that might be the case, but we really just don't know enough about it yet to know how to control this and how to optimize these connections. Then there's a question about stability. Do all the different approaches make something happen that's going to last as long as another approach? Does one better than the other in the long term? In general, patients don't want to go through something and come back down and come again and be back and forth. you want to consider the stability of the approach as you're picking it. Another issue in the brain is reversibility. If we do put in something and it's not working right, what can we do about it? And then there's these outcome measures that I referred to earlier. So do we have the right measures for TBI patients so that we could say that actually did something better than what their normal path would have been? To really know what's good, we have to have quantitative outcome measures for that patient and try to improve their overall functional response. For the military, a couple of special considerations with TBI. So TBI itself raises questions about ability to consent. So there are standard questions and procedures that anybody doing traumatic brain injury studies will ask a patient before they consider it a valid consent. When you don't have the ability to get a valid consent, then you have a surrogate consent. Now, if you're using NIH dollars, surrogate consent is very different than if you're using Department of Defense dollars, even if you're doing it at NIH. So the rules for the human subjects follow the dollars paying for the research. So for the DoD, you actually have to have, if you're using surrogate consent, you actually have to have a potential benefit for that individual patient. Sometimes if it's a very low risk, you're just going to take an MRI or something of them, and that's a very low risk. And if you get a clinical read of that, it may be a benefit because that's not standard of care. So there's some things that are fairly, easy to think about. That could be okay. But it all depends on the Human Subjects Committee and what's going on with that patient. So it's actually quite, different to think about when you're talking about benefit to a patient individually versus a class of patients globally. That's a very different context to have to work through. The military is doing really well, at least in our situation, of partnering with the civilian clinical studies together, and especially, as I mentioned, for these acute stage of research, because there's a lot of challenges to doing studies in theater, where that patient would be getting maybe the initial injury, and in general, like if you're doing something with neuroprotective strategies, you want to get in there right away as quickly as you can and treat that patient before the secondary consequences that cause you to lose more cells. And then there's these studies I mentioned in this natural history and outcome measures. These actually have a fairly good advantage within the military healthcare system because those patients are more likely to stay in contact, and they're going to stay in the system in terms of the healthcare that's offered to them, and they're a more likely population to just continue the study and have reason to be coming back to those doctors. They might be transferred around a lot, but in some cases you can still work with that depending on what's going on. Civilian populations are much more likely, especially for something like a mild TBI. So many are going to think, oh, I'm sort of done with it. It's okay. I went in, I got checked. They said I could go play again. And are they going to continue coming back? We wanted to come back 3 months, 6 months, a year, up to 5 years for one of the studies we're doing. The ones that are going to start to develop chronic symptoms and think that something might not be so good, maybe they're the ones that come back and the others don't, right? Because they're fine. That's going to bias your study. So we have to deal with that and acknowledge that and know how to work with it for the civilians. And that's true around the country for everybody that's dealing with civilian populations. So I'm going to end there. If you have anybody that is interested in the center or the clinical trials or participating in a clinical trial, we have a site that they can go to that will refer them to any of our studies. We have about 23 clinical studies going on, about 30-something preclinical animal studies, and 17 of those are actively enrolling, I think, of the clinical studies for patients. So I'll just stop there and answer any questions and hope I haven't gone on too long.
You mentioned the complicated interplay between the different types of injury. Could you explain a little further, like what is meant by a contusion versus hemorrhage? Like what are those differences?
It depends on what the blood does, basically, which layer it's in, and how the hematomas versus subdural or arachnoid, subdural and arachnoid, subarachnoid. So it's really the layering and how the blood accumulates, kind of thing, for all of the blood-related ones that I mentioned. Okay.
In the severe blast injuries, what comorbidities are there for damage to the lung? What percentage of the severe?
Right. So for blast, there's been a theory, which I think was originally by Ivy Cernak when she was at Georgetown and now she's at Hopkins, about the blast hitting the lung and making vascular changes up through the brain as part of the injury. There's another, group that's really looked at the blast wave, energy wave itself coming and how that might, work through the brain directly. We really don't know exactly what's going on yet. So that's one of the things we want to do with the pathology. And until you get that, you won't know. So if you have pure blast injuries and you can actually do the pathology, which is what we brought Dan Pearl to do, then we'll know what are the consequences in the human brain of somebody who's had blast and not had other complications associated with it. The soldiers do have the, the vests and stuff. So, most of them, there was a concern for a while that I thought, it's not my field for that part, but I thought was played down, that the protective armor was making the effect worse on the impact, but I'm pretty sure that was found out that was not the case.
You talked about imaging and some new kind of equipment becoming available soon for the second time. Who is it? The company Science you mentioned?
Right. So the PET MR combined, the molecular MR is. I can probably find it without too much trouble. Skip some of that. So this is just coming online. This is actually the combined machine being installed at NIH from like a month ago. So this is the molecular MR, PET MRI. We're also doing something with GE they got funded through the NIH extramural program to develop a head-only magnet with the Mayo Clinic. So this is GE research, not GE Healthcare. And this isn't officially signed by us yet, so I can't talk about it too much, so I'll skip by fast. But this is all the picture. But they did give me permission to talk about it today. So what we're doing is we're trying to partner with them so that what it is it's a, in the long run, what the Mayo Clinic should test for FDA approval will be a magnet with 3 Tesla MRI, which normally would fill a big part of this room and cost millions of dollars in renovation as well as millions of dollars in magnet. And then it would have to have like venting out and stuff to keep it cooled. This would be something that would fit without any renovation, about the size of an office, and cost about less than $1 million, and could really go pretty much anywhere in a hospital, and should really change the access to MR in the field if it's successful. It will take years, a few years, like 5 years down the line, to even get one tested. But we're really excited about it for TBI because what we want to do is the guts of that smaller one, which actually doesn't require venting either, the guts of that smaller one, we're going to try to work with GE to sign. We haven't signed on the dotted line yet because there's been all this movement of hospitals. We're trying to work with them, and what the idea is that we'll take a big one prototype and we'll put in the smaller pieces and test it for traumatic brain injury specially, and develop new sequences for scanning that really pull out some of the MRI findings that are hard to do. And really, by having it head only, it's closer to the head, you get better signal to noise, and it's actually going to be more powerful than a normal 3 Tesla for the head-specific piece. So we think that that's going to be really good, but I don't have it signed on the dotted line yet. But that's how the Department of Defense, for us, the center, is partnering with the extramural NIH and GE Research and Mayo to try to bring that. We're hoping to get it to the soldiers and service members at Walter Reed Navy about 2 years before it goes to Mayo for all the civilian testing for FDA, because at FDA you need all the different ages and types of problems that it might encounter.
Great.
You mentioned patient-specific effects for TBI outcomes and
Right,
treatment
patient-based
response.
factors. Yeah.
Patient-based factors, right. And you mentioned genetics and epigenetics. Do you know to what extent have those been studied? Do we know anything? Or?
There's a lot of effort going on that. So across the country, the Neurology Institute of NIH has been behind developing what we call common data elements. So everybody could do certain things for TBI patients whenever they're doing clinical research. And one of that pieces of that is developing blood draws for patients when they come in so that people can do genetic analysis and expression analysis and protein analysis. And to some extent, that is very expensive in the genetics. The human subjects approvals are a little touchy about the genetics as well, and what the patients are or not going to get connected back to them. So it's a little bit harder to do than it sounds, but there's a lot of groups around the country trying to accumulate enough of the right cases and the right samples so that you can do these kind of studies. They take a lot of samples from patients. Yeah, like the genome-wide analysis and stuff. Those take thousands and thousands of cases to get statistical significance. And they have to be developed for, as best you can, to have them connected to the clinical information that helps you decide that heterogeneity problem, because if you mix a bunch of different types of injury together, your genetic analysis is wasted. Yeah. Go ahead.
So what's the next focus for your studies? You mentioned that stem cells weren't doing so well, or regenerative capabilities within the brain aren't doing so well as in other areas. So what's your next step? Are you still just trying to characterize the current subjects you have, like you said, with the MRI and things like that?
So we're doing it all in parallel. I didn't really talk about all the studies going on at our center because I was sort of asked to do a broader picture than that. So we have 6 research programs, and one of them is neuroregeneration. One's neuroplasticity, one's neuroprotection. So those all together are working. We have about a dozen different research projects in each of those, and we're all trying to do different approaches to overlap and collaborate to see what's going to work. Because I think you have to do them in concert. You have to try to save whatever cells you can save. You have to try to, stimulate the neuroregeneration or transplant in, and different people are doing each of the approaches that I mentioned. And you have to then try to get them to connect properly to make them functional. So we're actually doing some studies in each of those. Yeah. Some of them are animal studies and some of them are human studies. Yeah.
I wonder if you could talk a little bit about human subjects and especially kind of the interface with the neurobiology, which is what you're really focusing on, but also the fact that you're working with injured people, the psychiatric side of that. Is there interplay between neurobiology and psychiatric?
Yeah, I mean, that's what we want to figure out if we get the right assessments together. So there's a lot of assessments going on for the psychological, neurological, cognitive, emotional, all the domains. And really evaluating any given patient as much as we can. We call it the phenotyping core, which is really like, everything you can possibly imagine for some of the studies. Some were so long that we had to cut them down because nobody would volunteer for them, like 4 days of assessments. We're trying our best to connect them as best we can. So the problem is you have to do it non-invasively to connect to the neurobiology. So that's why the animal pieces are very helpful to try to, so for the human studies, I mentioned all the MR. We have similar MR and PET machines for the animals as well, and behavioral assessments for the animals. So we're trying as best we can to connect the neurobiology through that by having parallel studies.
I'm mindful of the time, and I know some of you need to move on. Perhaps Dr. Armstrong might be willing to take up some questions directly afterwards, but I wanted to thank everybody for your attention, and especially thank Dr. Armstrong for a very informative presentation. Thank you very much.